Great. Thank you, Ramnik, and thank you so much for the opportunity to speak here today. And I'll tell you about our ongoing work at the Klarman Cell Observatory at the Broad Institute, where we are actually working to generate cell atlases, and we really use these as roadmaps to understand tumors. So in today's talk, I'll sort of divide it into 3 phases. In the first one, I'll give you a brief intro for single-cell genomics, and then I'll do a deep dive into one example of what we can learn when we use these methods on tumors. And then I will talk about the needed pipelines for tumor atlases. These are pipelines that we built throughout the last few years, and then I'll tell you about our efforts towards a human tumor atlas. Okay, so I will start with the intro and one story. So we all know that tumors are very complex cellular ecosystems. They're made out of malignant and non-malignant cells. For example, they have a lot of immune cells, they have fibroblasts, they have endothelial cells, and so forth. And this complexity has been a major challenge for functional genomics as well as precision medicine. But recent advances in single-cell genomics, specifically single-cell RNA sequencing, as well as spatial genomics, now put the dissection of such complex ecosystems within our reach. Just for an example, single-cell RNA sequencing, when you use it now at the Broad, we can profile tens of thousands of cells in one run in very, very short time. So if you can see on the graph on the right, actually, you can see that in 2012, actually this is a paper from Rahul Satija, who's sitting here in the audience, and Alex Shelik will be here tomorrow. We profiled 18 cells, and we thought that we were at the top of the world, and we were super happy. But by now, we've profiled more than 19 million cells at the Broad. And I don't have statistics for other centers across the world, but I'm pretty sure that we can see a very, very similar trend. So how can these methods actually help us understand complex ecosystems like tumors? Well, they can tell us which cells are in the tumor, which molecules these cells actually express, which other cells do these cells talk to? What are their partners? What is the functional effect of this interaction? What happens as a result? Where these cells are spatially located in the tumor, which is very important. And ultimately, what will happen clinically? What will happen when we add a drug or intervene in some way or the other? So using these methods in the past several years, we've gained insights into several different tumor types, including gliomas, ovarian cancer, head and neck, and so forth. And today I want to give you just one example, which is a study. It's a very recent study by Livnat Jerbi-Arnon and Ben Izar. And we all know that checkpoint inhibitors are making a tremendous impact now for cancer patients in the clinic, and we see durable responses for many patients and several different tumor types, including melanoma. This study is around melanoma. But there's also a major challenge that we actually have to face, which is that some of these patients are either resistant from the get-go to these immunotherapies, or they develop resistance with time. So one hypothesis is actually that the tumors that are resistant are what we called cold tumors. They don't have immune cells coming into the tumors, while the tumors that are responsive are what we called hot tumors, and they are infiltrated with immune cells. So to gain some sort of insight into this question and to start to understand the mechanisms underlying it, what Livnat and Ben did is take a cohort of melanoma patients, and here we had around 31 melanoma patients, and about half of them were resistant after treatment, and 15 of them were untreated. And what we did is we performed single-cell RNA sequencing from all these tumors from the patients, and we also used the bulk RNA sequencing results that we had from the TCGA. This is a very big cohort of samples. And when we did that, what we actually see is that the malignant cells express what we call an exclusion program. What happens is that this program is responsible for exclusion of T cells from the tumor. So when it's induced, there are no T cells coming in. And when it's repressed, T cells can come in. And you can see the signature here. So when we looked at the signature more closely, we see that the genes that are induced are MYC targets and CDK4 and CDK4 and 6 targets, additional targets. The repressed part of the signature includes MHC1, interferon gamma, and senescence-associated proteins. And what was nice about it is that when we actually look at this program, it separates the untreated and post-treatment patients. And we did this completely independently from the exclusion up or down. And I think this is a very nice result. And these two signatures, the treatment and the exclusion, were very, very similar. So we actually put those two signatures together, and we call this the resistance signature. So if what I'm telling you is true, we might be able to see this spatially within a tumor, right? We can see malignant cells that express the resistance signature in a high level. We don't really expect them to be close to immune cells or T cells, while the ones that repress it, we actually would expect to see next to immune cells. So we tested this using a multiplex spatial assay developed by Peter Sorger. And what you can see here, you can see the T cells are in blue. The malignant cells that have the resistance down signature are in red. And if you look closely, you can see that they're actually very close to T cells, while the malignant cells that have the resistance up signature here in green seem to be in sort of like set-alone deserts without any immune cells. And this is just one tumor where I can show you this example, but we did it across many of the tumor samples that we had, and we see a very similar picture. So what's the importance of this program or signature that we see? Does it have any sort of clinical prediction power? So in order to do that, what we did is we looked at an independent cohort of patients which were treated with PD-1 and CTLA-4, and a biopsy was taken before treatment, and these patients were monitored throughout their disease. And what we could see is that actually this program is associated with could give us the outcome of the progression-free survival, and it could also predict clinical response and also the clinical duration. So great, we have this program, but wouldn't it be amazing if we were actually able to reverse this program somehow and then maybe treat with immunotherapy? So the combination of these two therapies together might really help the patients that are resistant. So if you remembered when I showed you the signature, we saw that there are CD4, 4, and 6 inhibitors that are part of the signature. And currently in the clinic, there is a drug that can target CDK4 and 6. It's mostly used for breast cancer patients. But here we decided to test this and see if it could reverse our signature. So what we did is we took melanoma cell lines that actually express high level of the signature, and we either had cell lines with and without drug, and then we performed single-cell RNA sequencing. And what you can see here is actually that initially there are about 10% of resistant cells when you add the drug, which is Abemaciclib. You can see that the number of resistant cells drops dramatically, and also the cells that remain within the dish become senescent, which is part of the down signature, right? So it's part of the SASP pathway. So it's really nice. We see this in vitro, but do we actually see this in vivo? And the answer to that is yes. We use a B16 melanoma transplantable model, and this model is actually does not respond to any type of immunotherapy. And what we did is we used combination therapy of Abemaciclib plus IO, and what you can see is that the mice that got the treatment in a specific sequence have a very, very good clinical benefit from that. And now we're moving towards planning of clinical trials at the Farber to try and test these combinations. So just to summarize, I showed you that we can use single-cell genomics to find a malignant cell program that's associated with cold and hot tumors. This program has a predictive value in independent cohorts, and we can actually start now to reverse this program by using CDK4 and 6 inhibitors. Okay, so this is just one example. Initially, I showed you papers from different tumors as well. There's glioma, head and neck, and so forth. But how can we make this into something that's systematic? How can we make systematic pipelines for tumor atlases? Well, first of all, we need to overcome several challenges. So this is sort of a modular process, right? We need to get patient recruitment and consent in the hospital. We need to be able to process all types of samples. So there's resections, there are biopsy, which are much smaller, there's ascites, all kinds of needle aspirates and so forth, pleural effusions. We need to come up with a good experimental design to actually make an atlas. We need to have good experimental tools to get the measurements, and hand in hand with that, we also need good computational tools. So I think the first thing that we did was to try to have single-cell RNA sequencing working on fresh sample, meaning you get a sample from the hospital, from the OR and the IR as quickly as possible to the broad, process the tissue, dissociate the tissue into its components, and perform single-cell RNA sequencing. And throughout the last several years, we've built really robust pipelines, and here you can see several examples of such tumors that were dissociated fresh. And overall, we analyzed around 20 different tumor types. Okay, so this is sort of a not difficult, but a challenging process, right? You have to wait for the sample to come from the OR, you have to be ready, you have to dissociate, you can do one sample at a time, and so forth. So we really wanted to be able to actually process frozen samples. And so in order to do that, Naomi Habib, who's a postdoc, joint postdoc between Aviv Regev and Feng Zhang, she invented the method called single nucleus RNA sequencing, and it's a method that instead of looking at the cells, can now look at the nuclei, and you can use that on frozen tissue. So this method has several advantages. First of all, it doesn't require cell dissociation. So if, for example, you have those finicky epithelial cells in your tumor, you don't need to dissociate. Dissociation hurts epithelial cells quite a lot. You can use frozen or lightly fixed tissue. An important thing is that this actually decouples the collection of the sample from the processing. So you can collect a lot of samples, freeze them, and then process them together. Importantly, this really allows you to access banked samples. So if you have a rare glioma tumor in Spain and one in Germany and one in MGH, you can still bring all these samples together and process them in a very similar format. It also allows early pooling within and across donors, and you can do it on a massively parallel scale similar to single-cell RNA sequencing. So you can run tens of thousands of nuclei. So this was all great, and Naomi first worked on the brain. A lot of methods people work well on the brain. Brain cells have a lot of RNA, so sometimes it's easy. So we used this method on tumors, and one of the first tumors that we actually tested was neuroblastoma. And as you can see, we weren't very successful at all using this method on these tumor types. So luckily for us, we had an excellent postdoc, Eugene, who decided to actually make this work. So he tested several different conditions, over 104 different preparations that were sequenced, meaning he did a lot more, where he changed the mechanical stress, the chemical composition, including the buffers and the detergents. And at the end of this process, which took around a year, he came up with what we call a single nucleus RNA sequencing toolbox, which actually allows you to find the conditions pretty quickly that work for your tissue. Okay, so we wanted to, of course, test this on the neuroblastoma, which over a year we've been trying to work for and get working. And here you can see what we call the after results. And you can see that we found a buffer that works great for neuroblastoma. And actually, from the before and after we used the toolbox, it took us only 4 weeks to get these great results. And this toolbox has been very effective for many different tumor types, and you can see a few examples here. Okay, great. So we have very good tools for single cell and single nucleus RNA sequencing. So we can look at the 3' at the 5'. Alex is going to talk about nanowells tomorrow in more detail, and he will also talk about mutation calling, I'm guessing, from these from cells. We can also now look at the TCRs and BCRs. For example, if we're interested in immunotherapy, it's actually very important for us to follow these. We also really care about spatial analysis, and we're working now pretty hard and diligently to bring in spatial methods. And the idea here is to perform multiple measurements on the tissue, both on the RNA and on the protein level. For the protein, we use Codex and MIBI, and we now use the GeoMax and SciSafe. I showed you the example for melanoma. And for RNA, we're working together with Xiaowei Zhang to work on MERFISH, and with Fei Chen and Evan McCosco, and Ramnik showed this before, we're trying to implement SLIDE-seq. In addition, we also want to look at the chromatin states in single cells, so we're now working to bring single-cell ATAC-seq as well, and try to look at these tumors. And we can also do actually lineage tracing to look at sort of evolution by looking at the mitochondrial reads. We can also start looking at protein. I don't know if Rahul's going to talk about SITE-seq or not, but it's a way to look at cell surface proteins together with your single-cell RNA sequencing. And importantly, we're also starting to deploy methods for multiplexing of samples. And this has several advantages. The first one is that it reduces the cost because you can load more samples per your droplet channel. And the other one is that it reduces your biases because you can process several samples at the same time. And in terms of multiplexing, we're working on several approaches. One is the genetic approach, where you actually just demultiplex by genetics. And the other one, which Rahul might talk about tomorrow, is using antibodies with oligo tags that could also help you demultiplex your samples. So we've worked really diligently on workflows. I know it doesn't sound like the most exciting thing in the world, but actually it's very important when you work together with hospitals, and we work very closely with Dana-Farber Cancer Institute as well as MGH. And this process starts with patient identification and consent. Then there's the biospecimen acquisition and the allocation of the biospecimens to the different assays. And at the end, we finish up with the data analysis and processing, and we get the results. So here you can see one example for workflow for breast cancer. This is for prospective samples. And the idea here is that we actually need to do things very quickly. So since the moment that the tumor leaves the body, we try to expedite everything in order to get it to profiling. And actually, the hospital team works very closely together with the Broad team in order to do that. And we have a dedicated, what we call Slack channels, and these are very good for quick communications and coordination between the clinical and the research sites. You can see here an example of a lung tumor that we process and all the conversations that happen. When is the tumor coming? When are we processing? And so forth. We also have very good computational pipelines. So Boalia, a postdoc in our lab, has now generated a cloud-based shareable and reproducible and very, very fast pipelines. So you can analyze around 1 million cells in 2 hours instead of days. One of our main objectives is actually to disseminate our SOPs. So in terms of experimental protocols, we put everything on what's called protocols.io. It's a repository for protocols. It's open access. And the computational workflows and protocols and pipelines all go on GitHub and Docker. One last thing about this effort is that we have what we call the SWAT team. Maybe it's not the best name, but the idea here is that the role of this team is to actually share our knowledge and experience in working towards generating tumor cell atlases. So what does that mean? It means that we put together a team which would have a research scientist that knows how to do all the single-cell RNA sequencing method, hopefully soon the spatial methods, computational person, research associate or PDA that knows how to process. And this team goes to different places, to a different hospital, for example. Our first visit was to St. Jude. And the idea there is that the two teams meet. They talk about how we do single-cell or single-nucleus RNA sequencing. And importantly, they perform and do a side-by-side comparison where our team teaches them how and what we do, all our know-how. And this turns out to be very interesting because different hospitals have different workflows, and actually we can learn quite a lot from that. And here you can see an example of one of the SWAT team visits to St. Jude. So importantly, we want to use single-cell or single-nucleus RNA sequencing within the clinical arena, right? So now, when a metastatic breast cancer patient comes into Dana-Farber Cancer Institute and they take cores in the IR, there's a core that goes to pathology, a core that goes to whole exome and bulk RNA sequencing, but also 1 or 2 cores come to us for single-cell RNA sequencing. And here you can see a few examples of metastatic breast cancer lesions, and these are biopsies to different places in the body, to the liver, to the bone, lymph node, and breast. And then you can see the composition of the tumor microenvironment. You can look at the epithelial cells, the cancer cells, and you can look at the interactions between them. And importantly, you can then make connections to different clinical features that are associated with these samples and tumors. Okay. So how can we actually use all these tools and these methods and workflows and protocols to actually begin to generate a human tumor atlas? Well, first, what do we want in such an atlas? What are the cross-cutting things that we want? Well, we want, adult and pediatric samples. We want to have samples or tumors from both genders. We want to have diversity. We want to look at different types and subtypes of tumors. If you look at the TCGA website, it says that there are around 200 different types of tumors and many more subtypes. So there are solid tumors, non-solid tumors. You want to be able to look at transitions. So we want to look at pre-malignant, malignant, and metastatic. You want to look at response, and then resistance, intrinsic or acquired. I'm guessing it will also be very interesting to look at environmental factors and how they actually affect these tumors. And then you need to know how many patients, how many cells, how many nuclei, how many fields of view if you're doing spatial analysis that you actually need to look at, which methods to use. And hopefully at the end we'll be able to compose tumor atlases, and that will help us generate better drugs for precision medicine. So in the last year or so, we've been working at the Broad to come up with new methodologies for building these atlases, and they encompass several different molecular measurements as well as spatial measurements. And we're working very hard to come up with new computational methods that can help us put together the single-cell RNA or single-nucleus RNA sequencing data, because these are from dissociated tissues together with the spatial analysis. And we have two pretty large ongoing efforts. These efforts are part of the NCI Moonshot project. The first one is called the Human Tumor Atlas Pilot Project, and it focuses on generating atlases of metastatic breast cancer and neuroblastoma. And in addition, we're developing experimental and computational methods for 5 additional tumor types. And we also have what we call the Human Tumor Atlas Network, which actually focuses on understanding resistance to therapies. So for metastatic breast cancer, we're focusing on CDK4 and 6 inhibitors, and in colorectal cancer on immunotherapy on MSI patients. And in melanoma also, we're looking at immunotherapy. And so we hope that these type of activities where we build tumor cell atlases can actually help us better understand tumor biology, help us make better drugs, better diagnostics, come up with new biomarkers that are helpful, new clinical trials, and hopefully at the end of the day will help us come to an era of precision or personalized medicine. And so there are many people to thank. A lot of people are involved in this effort. I want to particularly thank Aviv Regev, who I work very closely with and is an amazing mentor and scientist, and you'll hear her give a talk tomorrow about the Human Cell Atlas. And a special thank you to all the patients and their families. And thank you for listening.
Detailed outline of how what you might be able to do for cancers. Many of the same principles could be applied to understanding how the environment might influence health and disease. And so, questions for Orit?
Norb Kaminsky, Michigan State University. That was just a fascinating talk. I'm curious, I'm really fascinated by your working so closely with the clinicians, and I'm curious, what criteria ultimately do you use to decide which patients you're going to sample? Obviously, this is a lot of work, very costly, and I know you mentioned a few different tumor types, but can you tell us a little bit more about that process?
Sure. So initially, when we started this process, I didn't show you our first melanoma paper, but that was a hodgepodge of patients. So whichever patients would come in, we would profile. And that was very important for us to start establishing the connections with the clinical fellows. You have to have really good connections with the clinical, oncology fellow, with the surgeon, and with all the hospital staff in order to actually get this to work. So that was sort of our first criteria. We're now moving to more sort of structured criteria to choose patients. We actually, to be honest, we never turn a patient sample down, to be quite honest. Like if there's a patient, patient sample, we almost always run it. Which is not very financially smart, but we view these as very, very precious samples. But in our projects now, we have a few criteria, especially on the H10, where we're looking at specific treatments, for example, for cancer. And sometimes you get a sample and it doesn't look good. You can do a few QCs before you run, and then you won't run it. So we're still quite in the mode of running whatever sample we get, but in some cases we have specific questions. You can say, for example, for lung, I want to profile only people who are smokers, X amount of people who are smokers, and X amount of people who are non-smokers. And as we move more towards working more with clinical trials, then you can take pre, post, or on treatments to more structured.
I could just ask one more question.
Sure.
Grab the mic here. I was really fascinated also by the information you gave us on the resistant and non-resistant tumors, and you gave us a little bit of an inkling of the differences in gene expression profiling. But I'm curious, what do you think actually is involved in those T cells not wanting to come into that tissue? I saw you had IL-6, interferon gamma on there. Do you think those are cytokines that are being produced, or is it PD-1 ligand or ligand 2 that's expressed?
It's the $1,000 million question.
Okay.
I don't know yet. Actually, here it's not exactly to do with PD-1. But I think there's more than one mechanism, and we actually just need to have a more structured study in order to gain more insight into this, where we do pre, on, and post-treatment. I think would be very insightful.
Thank you.
Ken Buckelheim, Brown University. I was really interested in the single-cell-seq, single-nucleus-seq relationship, and if you could speak to the comparability of those samples and information density and repeatability, I guess, because I can see just tremendous advantage to the freezing the samples down approach and then batch processing kind of thing. And then also whether all of those things apply to non-tumor cells equally as to tumor cells.
So first of all, there are just some samples that are frozen, and there's no other way for you to profile them. So I think that's one. The second is that when you use, for example, when you look at adipose tissue and you do single cell RNA sequencing, you get all the cell types apart from adipose cells, because these are big fatty cells, and you just don't get them, and you get them by the nuclei. So there's a little bit of trade-off between doing nuclei versus cells. You might get a little bit different subsets, but overall the picture is the same. The number of genes is a little bit higher in cells versus nuclei, but overall you get pretty much the same structure. If you look at the cell types, there are a little bit biases in each method, and that's why we tend to use both bulk RNA sequencing to figure out what are all the cell types in the tumor, as well as spatial analysis now to figure out if we're missing anything by using one of these methods. But when you do nuclei, you get pretty much all the cell types. You get cell types that you don't see in the single cell RNA sequencing. For example, in the gut, there is at least one cell type that we couldn't see when we just did the atlas of the single cells, and we can see it with the nuclei. And yes, we actually have a really big effort to look at different tissues across the body, and we leverage what's called the GTEx project. It's a NIH project that was looking at the different tissues across the body and the expression, and then connecting it to genetic variation. And we were able to leverage some of these samples, and using this toolbox, we actually showed that we can process using single nucleus RNA sequencing at least 8 of these tissues, and we're now extending this to other tissues in the human body, and this is part of our work for the human cell atlas that Aviv will talk about tomorrow in detail.
Thank you.
Mike Mancini, Baylor College of Medicine. You talked about maybe it's not so exciting working on protocols and workflows, but what about the antibodies that are now being used We just hosted a big conference on imaging and multiplexing and had all the big vendors in, and none of them are really qualifying antibodies. So how do you guys know that the antibodies that are now being put through all this are really specific? There's a tremendous amount of junk out there, as I'm sure we all know.
Yeah, so I think working with antibodies is always hazardous, and you need to try to figure out what are good antibodies to work. I don't know if Rahul is going to talk about that tomorrow, more about site, when he's talking about site-seq, if he's talking about that at all. But there are different ways to validate antibodies. For the MIBI and the Codex, we work very closely with Gary Nolan, who has different ways to validate his antibodies. So one of the things that they do is they use, for example, cell lines that express or don't express a specific antibody, and they can see the different staining. I think that's one option. The other option is, of course, to knock out that specific gene and then do staining with the antibody. I think there are more antibodies that work for FFPEs versus necessarily frozen tissue. But that's an art to itself, and this is why we're using different methods to look at multiplex RNA, which, you know, it's not protein, but RNA gives us a lot of information. So if you put together the single cell, the single nucleus RNA sequencing together with the multiplex RNA methods, which don't have the problem of antibodies, together with antibody methods, I think the combination could be very helpful.
So, Orit, there is one question from the online community. So the question is, what is the possibility that you are selecting only for a subset of cells within a tissue or a tumor?
So that's always a possibility. As I said, every method has its own biases. So epithelial cells don't like to be dissociated. So sometimes you see them better in when you do single nucleus RNA sequencing. But this is why we actually try to put a few methods together. So we do the bulk RNA sequencing, the single cell or single nucleus sequencing, as well as spatial methods. And we hope that bringing all these methods together can actually tell us more about the accuracy of cell types and cell proportions within a tumor or tissue.
Any other questions for Orit?
Could you comment on the similarity and the differences between, using the fresh tissue to do single cell RNA-seq versus using the prefixed to do single nucleus sequencing? How similar the gene expression profiles are besides the cell population?
So you get different genes when you do these methods, but overall the cell types that you can call out of these are very similar at the end of the day. You miss some when you do fresh, and you might miss some when you do nuclei, but overall it's pretty comparable.
As I understand it, nuclei sequencing gives you premature
so
MRI, right?
So you look at that also. So when you do the computational pipelines, you include the introns in them. So you have a different computational pipeline to use for that.
How accurate if you use that tool to kind of estimate the mature MRI to make it comparable? I mean, basically I was thinking maybe single nuclei sequencing is a surrogate for, normal type of single cell RNA-seq. Is it the fidelity is high enough that you can really use this as a surrogate method because of the limitation of multiple sites to collect samples?
In some cases, you can detect cell types that you cannot detect in the single cells. So yes.
Okay, so maybe we should move on. Thank you, Orit, again for that nice overview on single cell methodologies with tumors.